Assalamu alaikum dear students. Today we will discuss lecture 1, which is microorganisms. In this tutorial we will discuss what are microorganisms and also discuss about the evolution of microorganisms. So moving toward the first slide. What are microorganisms? When we simply define them, a microorganism is an organism which is microscopic in nature and can small enough cannot be seen with the unaided human eye. So the basically study of microorganism is called as microbiology, and under the term microorganism we include bacteria, fungi, archaea, protists and viruses. Now moving toward the next, evolution of microorganisms. When we talk about the evolution of microorganisms came into being on Earth over the period of 1.2 to 1.5 billion years ago. And actually, the fossil microbes have been found in rocks since 3.3 to 3.5 billion years old, which is very earlier. And the microorganisms have principal task of recycling the organic matter in the environment. So we can say that they are absolutely essential to the health of Earth, and without the microorganisms, the Earth would be a permanent waste dump. This shows the importance of microorganisms. There are two theories about the evolution of microorganisms. The first theory is endosymbiosis, which is the condition of living within the body or the cell of another organism. Whereas second is panspermia, which is the hypothesis that the microorganisms may transient life from outer space to habitable bodies, just as the process of transmission. So when we talk about the scientific evidences that shows the life began on Earth some 3.5 billion years ago, they say that the life have evolved into a variety of forms, under which biologists then classified into the different hierarchy of taxa. Some older cells on the Earth are basically the single-celled organisms. We call them archaea and bacteria, whereas when we study according to fossil record, it indicates that mounds of bacteria once covered the Earth. Then they start making their own food using the carbon dioxide in the atmosphere, energy harvest from the sun. So this process called photosynthesis produced after enough energy to change the Earth atmosphere totally. So this means that the at start the bacteria cover, and then with the changing in atmosphere, they are continuously changed in Earth atmosphere. After that, when oxygen was discovered and the atmosphere was changed, the after them new oxygen breathing life forms came into the Earth, and with a population of increasing diverse bacterial forms, the stage was set for more life to form. So there is evidence that mitochondria and chloroplasts were basically primitive bacterial cells, and this evidence is also supported or described in endosymbiotic theory. Whereas, because we know that symbiosis occurs when two different species benefit from living and working together. So when one organism actually lives inside the other, it is called endosymbiosis, that we will discuss in previous. So the endosymbiotic theory describes how large host cell and ingested bacteria could easily become dependent on one another for survival and results in the uh, permanent relationship, which is the important Then, over millions of years of evolution, the mitochondria and chloroplast have become more specialized, and today they cannot live outside the cell. These mitochondria and chloroplast have striking similarities to bacterial cells because they have their own DNA, and other is which is separate from the DNA found in the nucleus of the cell. So they have their own, and both organelles use their DNA to produce many proteins and enzymes which are required for their function. So a double membrane which is surrounded both which is surrounding both mitochondria and chloroplast is further evidence that, and each was ingested by primitive host, and the two organelles also produce like bacteria replicating their own DNA and also directing their own division. So a mitochondrial DNA has a unique pattern of inheritance which is passed down directly from mother to child as it accumulates the changes much more slowly than other types of DNA. So, because of the unique characteristics, we can say that the mitochondrial DNA has provided important clue about the evolutionary history. For example, we can say that the difference in mitochondrial DNA, which is examined to estimate how closely related one species is to another species. So, after that, the condition on Earth 4 billion years ago were very different than those are currently there are conditions. So the atmosphere, because we know that at start the atmosphere have no oxygen and ozone layer did not yet protect it from harmful radiations. Heavy rains, lightnings, volcanic activities are very common. So the earliest cells which are originated in this extreme environment are extremophiles, and these are extremophile archaea that have ability to survive in extreme habitats. So astrobiologists are now using these archaea to study the origin of life on Earth and other planets because these are present at that time. So because archaea inhabit the places previously considered incompatible with the life, so we can say that they provide very important clues that it will improve our ability to detect the extraterrestrial lives. And similarly, current research also suggests archaea, which is capable of space travel by meteorite. Such an event termed panspermia could result in the seeded life on Earth or elsewhere. So we know that after the presence of archaea and bacteria changed the earth dramatically, they will establish a stable atmosphere and produced oxygen in such quantity that will eventually different life forms could evolve as a result, and that needs inner oxygen. So the new atmospheric conditions which are extremes where the conditions are not as extreme as previous, the life had created the conditions for new lives to be formed, and this process is one of the great wonder of nature. We know that life is on Earth is famous for its diversity, and when we talk about throughout the world, we can find many millions of different forms of life. Biological, according to biological classification, which helps to identify each form according to their similarities using a set of rules and estimate how to closely related it is to certain common ancestor in a way to create an order. So by learning to recognize these certain patterns and then classify them into specific groups, the biologists are better able to understand the relationship that exists among the variety of living forms that are present in the planet. Though the first and largest and most inclusive group under the microorganism category are classified as domain, which has specific three subgroups: bacteria, archaea, eukarya. The first group defines whether an organism is a prokaryote or eukaryote. So the domain was proposed by a microbiologist and physicist named as Carl Busse in 1978 and is based on identifying the similarities in the ribosomal RNA sequences of the microorganisms. So we will discuss one by one. Whereas the second largest group is kingdom, and basically 5 major kingdoms have been described today, and includes the prokaryota, which include archaea and bacteria, protoctista, protozoa and algae, fungi, plantae and malia. And the kingdom is further split into the division, class, order, family, genus, and at the end is species, which is the smallest group. So the all this is this classifying units, and the science of classifying organism is called as taxonomy, and the group making up the classification are called as taxa. So taxonomy basically consists of classifying the new organisms or reclassifying the existing one. Microorganisms are basically scientifically recognized using a binomial nomenclature using the two words that refers to the genus and the species. So the name which is assigned to microorganism are in Latin word, and the first letter of the genus name is always capitalized, whereas second is short. So classification of microorganism has been added by the study of fossil and recently by DNA sequencing. Now the methods of classification are constantly changing, and most widely employed methods which are used for classifying the microbes are based on morphological characteristics, differential staining methods, biochemical testing, DNA fingerprinting, or maybe DNA base composition, PCR and DNA chip. These are the basic methods that are now used for classifying the microorganisms. So the first microorganism that we will discuss is bacteria. Bacteria are basically single cell microbes, and when we talk about the cell structure is simpler than that of other microorganisms, as bacteria have no nucleus and membrane bound organelles. So inside their control center, which is containing the genetic information, it contains a single loop of DNA. Some bacteria have extra cell circle of genetic material which is called as plasmid, which often carries genes that give the bacteria some advantage over the other bacteria. So we can, for example, we can say that the plasmid contain a gene that makes the bacteria resistant to certain antibiotics. The next is that we will discuss is viruses. Viruses are basically smallest of all microbes, and one estimation about their size is they are so small that 500 million rhinoviruses, which cause the common cold, could fit on to the head of the pin. So they are also unique because they only alive and able to multiply inside the cell of other living things. So the cells then multiply under the multiply is called as host cell, and basically the virus is made up of core of genetic material which is either DNA or RNA, and this core is then protected surrounded by a protective coat which we called as capsid, and this capsid is basically made up of proteins. So sometimes the capsid is also surrounded by additional spiking coat which is called as envelope. So enveloped viruses and non-enveloped viruses. So viruses are capable of latching onto the host cell and catching them inside them. These are some important characteristics of viruses. Now we will discuss about what are fungi and their. Fungi basically single cell or very complex multicellular organisms. They are found in any habitat, mostly live on the land, mainly in soil or plant material rather in sea or fresh water. So basically these are the decomposers group that grow in the soil or dead plant matter, where they play very important role in the recycling of carbon and other elements. Some are parasites also, which are causing diseases such as rust, mildews, and in crops basically the fungal diseases can lead to the significant loss for the farmers and agriculture demand. A very small number of fungi cause disease in animals, whereas in human these fungi cause skin diseases such as athlete foot, ringworm, and thrush. These are common some diseases. And now we will moving toward the protozoa. Protozoa basically are single-celled organisms. They come in many different shapes and sizes, which are range from amoeba, which can change its shape, to paramecium, with its fixed shape and complex structures. They are basically lived in a wide variety of moist habitat, including the freshwater, marine environment, and the algae. Next is algae can exist as a single cell, or an example is algae is Chlamydomonas, which then join together in chains, which is spirogyra, and made up of many cells. So, red seaweed example is. Then the last one is archaea. Archaea can be spherical, they may be slot, spiral, lobed, rectangular, or irregular in shape. Basically, they are unusual flat, square-shaped species which is lived in salty pools and also first discovered from salty pools. Some exist as single cells, whereas other are from filaments and clusters. And until 1970, this group of microbes was also classified under the category of bacteria. This is all about the microorganisms, brief discovery about their discovery, brief discussion, and then one by one, what are their important, some important characters. Thank you very much.